everyone, welcome to Bio Practify. I am Khushi and today we'll learn the biological databases in bioinformatics. Every single day, scientists upload millions of new data sequences, protein structures and genetic variations into massive online databases. They aren't dusty archives, they are living, breathing tools, helping us fight diseases, explore evolution and understand life at the molecular level. Imagine this, in the time it takes for you to watch this video, thousands of new genetic discoveries will be stored online, ready for scientists anywhere to use. Today we are diving into the same world of biological databases, what they are, how they work, key examples and how you can explore them yourself. So let's start. In modern biology, databases are the backbone of discovery. Without them, decoding the human genome would have taken decades. Vaccine development would slow down to crawl, and tracking new pathogens like COVID-19 variants would be nearly impossible. Think of them as a Google of biology. But instead of searching for cat videos, you're searching for the building blocks of life itself. Whether you're a student, a new researcher, or just curious, understanding these databases is like having a master key to unlock the age of bioinformatics. And if you could search one thing in the biological database right now, a gene, a protein or a disease, what would it be? Drop it in the comments. So what are the biological databases? It is a structured, organized and searchable collection of biological information. Think digital libraries for life sciences. They store DNA sequences, protein structure, genetic mutations, molecular pathways and even disease cases report. Their goal isn't just storage, they are designed for a quick retrieval and automated analysis so scientists can compare the DNA with the global pool and find answers faster than ever before. For example, if a researcher in India discovers a new bacterial strain, they can upload its genome and within hours, scientists in the US can download it and start developing a treatment. So now let's see what types of data are stored in these databases. First, genomic data like DNA and RNA sequences. Second, proteomic data, amino acid sequences and protein structures. Pathway data, metabolic and signaling networks, literature references, links to study and publication. And last, taxonomic data, classification of living organisms. Biological databases are vital tools for research, storing vast amounts of information in a structured, easily searchable format. Here are the main categories, each with a real-world examples what makes them unique. First is primary database. It is store raw, experimentally derived data directly from the laboratory research. No modification or interpretation. Examples are GeneBank, EMBL, and etc. Second, secondary databases contain processed, curated, and interpreted data based on the primary sources. Biologists add extra insights and annotation. For examples, UniProt or SwissProt. Third, composite databases integrate multiple datasets from primary and secondary sources for more comprehensive analysis, and it includes example NCBI or Ensembl. And last, specialized databases focus on narrow topics like enzymes, genetic disorders or metabolic pathways. Example includes KEGG and OMIM, genetic disorders. These categories prevent the redundancy and organize data efficiently and help researchers quickly find high quality, reliable information for any biological query. Now NCBI. NCBI stands for National Center for Biotechnology Information and it is the backbone of modern bioinformatics. It was established by Congress in 1988 to centralize and accelerate molecular biology research. It features GenBank, which includes DNA and RNA sequences from millions of organisms. Second, PubMed, massive databases of biomedical literature. Gene, detailed gene records. Plus, it is a powerful tool for sequence alignment and comparison. Taxonomy browser, complete classification of life. DPSNP, databases of genetic variants. And why it matters? Seamlessly links genetic data, literature, and curated information, so user can connect a single gene from a sequence to diseases, research papers, protein sequences, and evolutionary data. Let's do a quick example searching BRCA1 in NCBI. We can see its chromosome location, known function, sequence data, related research papers, and use BLAST to compare it with sequence from other species. The amount of detailed cross-link information here is staggering. By the way, the practical website navigation, searching for gene or protein in NCBI, UniProt and Ensembl will be covered separately in a dedicated YouTube Shorts video. So if you want an easy step-by-step walkthrough on how to explore these databases in real time, make sure you are subscribed. Next is UniProt. UniProt is the ultimate protein resources, a must-use for scientists studying protein structure and function. It is free, comprehensive and high-quality databases. It covers millions of protein sequences. And the key features of this are SwissProt, expert review entries, highly annotated, reliable data. Ensembl, it is annotated entries. It has rapid updates from new sequencing projects. Next, functional information. It has protein domains, post-translation modification and biological activity. 3D structure links, disease relevance and cross-references to our other databases. Produced by the UniProt consortium, EMBL, PBI, SIB, PIL. Interactivity. Map identifies and compares sequences across via API for large-scale studies. Next is Ensembl Genome Browser. It brings interactive, customizable navigation for hundreds of genome sequences. It has customizable track-based views, zoom from a whole journey to a single gene or nucleotide. It covers human key model organism, disease-related species, and its key features are visualization of exons, variants, regulatory regions, and comparatively genomes. Tools for exporting data in common formats, faster, PET, PCL, and a powerful API for computation access. Cross-species comparison, evolutionary gene tree, annotation of both coding and non-coding regions. It has rapid and free access, designed for researchers at any experience level, from student to professionals. Now let's jump into applications of biological databases. Biological databases have transformed science and medicine in ways you encounter every day. Evolutionary biology traces species' history, discover common ancestor, and analyze genetic divergence. Medical research, it detects mutation causing diseases like cancer or genetic disorder, accelerate drug design and personalized medicine, identify targets for new therapies based on protein structure and function. And the other application is outbreak tracing, rapid identification of pathogen, for example, COVID-19 variants, and monitoring their spread. Next, comparative genomics. It helps in studying model organisms, compare genes and proteins, develop animal models for human diseases. It also helps in synthetic biology / bioengineering. It helps in designing new proteins or genetic circuits for industrial, agriculture, or even pharmaceutical applications. So we saw what biological databases are and why they matter. Explored NCBI, UniProt, and Ensembl, and saw some application. Why they are, and its summary is that they are very essential tools. It is crucial for data storage, retrieval, and analysis. It helps in learning, constantly updated and requiring data, and it also helps. It is also a fundamental skill for any bioinformatician or molecular biologist. So these are the references that you can go through, and we are suggested few references that are NCBI tutorials, UniProt user guides, and Ensembl training materials. These sites can help you engage in the websites and can help you use the websites efficiently. So if this opened your eyes to the power of bioinformatics, hit that like button, subscribe, and stay tuned for upcoming videos. Thank you.